Okay, so welcome to the exam 3, I should say. I was going to say week 3. Exam 3 review session. I am just going to make sure I pop up the PowerPoint here. And we will start. So I did email this PowerPoint out to everybody. Actually, I'm going to leave it in the format like this because in case you want to type. So we're going to start with blood cultures. So we did talk about blood cultures within the last week or so. Again, blood cultures' whole purpose is to detect bacteremia or septicemia, essentially bacteria or organisms in the blood. So again, we can collect different sets depending on the physician order. So every set is going to contain an aerobic and an anaerobic bottle. So that's one set. So a lot of times we do 2 to 3 sets, and that just helps increase the sensitivity of detecting if there is an organism. Again, they should be collected approximately 15 minutes apart, ideally in a different location on the person. Again, you do have pediatric bottles. There's just one bottle in a pediatric bottle set, and that's around 1 to 3 mils that you put in that pediatric bottle. The anticoagulant, again, in the bottles is SPS. It helps just to keep that blood liquid. That's the purpose, again, of an anticoagulant. The only issue with SPS is it does inhibit a few different bacteria. We discussed those in the original PowerPoint, but it also inhibits all strains of Streptococcus anaerobius, so just keep that in mind. So once we draw these, we throw them into an incubator analyzer thing. It's going to keep them at a nice 35 to 37 degrees Celsius temp because that's what bacteria love to grow at. And the analyzer is continually scanning the bottles in there, and it's looking for the growth or detection of CO2, the presence, I should say, of CO2. So as bacteria are in the cultures, they will produce CO2, and that's what the analyzer is scanning for. When we get the analyzer dinging at us or beeping that there is a positive culture, we're going to remove it, take blood out of the culture, and plate it to everything, essentially. So your blood agar, your chocolate, your CNA or PEA, and your McConkey agar. So that is going to basically help grow pretty much anything that might be there. If it is the anaerobic bottle, you're also going to make sure that you set up an anaerobic plate. Now, the other thing you're going to do right away is gram stain. So besides plating those out and putting them in the incubator to grow, we're going to gram stain and report the gram stain results immediately to the physician. So there are some huge pathogens that we see commonly with blood cultures. Some of the ones are listed here, the gram-neg rods. It be any of the enterobacteria. You see a group like E. coli. The other one, big gram-neg rod, would be Pseudomonas. Gram-pos cocci, Staph aureus, Strep Pneumo, Strep pathogens. I mean, those are just pathogens we see all the time. And they're basically can be anything. Like I said in the original lecture, anything can really form a bacteremia if it has the opportunity. It's just we tend to see certain ones majority of the time. So contaminants, we will see contamination if we do not do a really good job cleansing to collect these. That is probably the most critical step in blood culture collection, is cleansing the skin really well. We do that dual cleanse where you first cleanse with isopropyl alcohol, and then you can go in with iodine over that. And we're also cleaning the tops of the blood culture bottles, all to prevent any normal flora off the skin from growing in the blood culture. We don't want to pick up normal flora and then have it be just nothing but a contaminant. So very common skin flora that we can see as contaminants will be your coag-neg staph. Probably the most prevalent one is going to be Staph epidermidis. Your diphthroids, which are basically your Crinibacterium rods that can show up. Now, if it's truly a pathogen, it will grow in all bottles or the majority of bottles. We're talking about these are growing in just 1 out of, say, 4 or 6 bottles. If it's only 1 bottle out of all the ones you collected and it's one of these, it's probably just normal flora. Yes, you can truly have Crinibacterium diphtheriae cause septicemia, but you should see more than 1 bottle show up positive. That's what we're referencing here. It's just 1 bottle showing up. So this is more skin flora then. Your bacillus species, except anthracis, of course, but all your other bacillus species tend to be normal flora. And then again, Propionibacterium acnes is a normal flora. Going into respiratory cultures, if it would click. There you go. So we discussed upper and lower respiratory type cultures. So we'll start here with the upper. My mouse is just being weird today. Okay. So different diseases in the upper respiratory world. We have croup, which is called laryngotracheal bronchitis, but that is most often caused by viruses, especially the para-influenza virus. The croup is that really working cough sound that children get. There's really nothing they get put on the account. Hepatitis, that does have a main cause. It can be caused by a few different bacteria, but the major one that it was Haemophilus influenzae type B. And then pharyngitis, of course, that is your strep throat, the main cause here. Again, there can be other things that cause it, but the main one, it would be your strep pyogenes. Remember, that's group A. Rhinitis is just common cold. We have our major viruses like the rhinovirus, coronavirus, and adenoviruses that all can cause colds. Stomatitis is due to herpes virus. And finally, thrush is inflammatory white patches within the mouth. So a lot of times caused by candida, which is a yeast. A lot of times you might hear this referred to as oral candidiasis, which is just thrush. Other diseases, periodontal, a lot of times caused by anaerobes. Any dental stuff could be nasty anaerobes. Or Eikenella corridans. We did discuss that with, that's housed in the mouth of people. And that's the one with, what do they call it, clenched fist wounds, when they go to punch somebody in the mouth and then they get the wound on their fist from it, and it can grow out Eikenella corridans. Remember, Eikenella is the one that smells like bleach. Parotitis is inflammation of your salivary glands. Mumps would be the big one there for the virus. Otherwise, bacteria-wise, Staph aureus. Whooping cough, that is caused by, again, Bordetella pertussis, which we all have a vaccine to. Augurs, there are some specialized augurs, remember, to grow Bordetella pertussis. So one is Bordet ginkgo. Remember that Bordet part looks just like Bordetella. Regan low would be another great one to list. And collection, typically, again, this is a nasopharyngeal collection. So it's kind of up the nose with a swab and twirl. If we see a membrane on the back of the tonsils, we have this question on the study guide. Some people always put epiglottitis, which is that little flap over the airway becoming inflamed. This is more referring to diphtheria. Diphtheria, remember, forms a membrane across the back, completely blocking the airway as well. So they both are similar and they both block the airway. But epiglottitis is just an inflammation of the epiglottis, little flappers thing. I don't always have to call it a flap. And then the diphtheria actually produces a membrane over the back. So that's the difference. So this, again, is Corynebacterium diphtheriae. You get the gist. Okay. And then normal flora in the mouth, upper respiratory area is a lot of times the viridin strep group. They are known to cause some dental stuff, but otherwise a lot of normal flora. Lower respiratory was going to be more bronchitises or pneumonias. Bronchiolitis, RSV, which is respiratory syncytial virus, is a huge cause. That's pretty much in children ages 2 to 5. Very commonly passed in daycares. Pneumonia, we have lots and lots and lots of pneumonia agents that cause it. So I'm not going to list them all. You should kind of already have an idea of all the different pneumonias. Some special mentions here, Mycoplasma pneumoniae, again, causes walking pneumonia, which is a little bit more of an atypical, milder kind of pneumonia. The major, most common cause of pneumonia is your Streptococcus pneumoniae. And then Legionella is mentioned here because, again, it causes Legionnaire's disease, which is a pneumonia-like illness. So there is an agar to grow this on, and it would be your BCYE agar, your buffered charcoal yeast extract agar. It's been a while since we talked Legionella. Sputums. We love to gather sputum anytime we think we do have a pneumonia, just because they should be coughing up all sorts of stuff. So sputum, again, is that mucus from deep down in the lungs, but we like to gram stain it immediately upon receiving it to make sure that it is a quality specimen. I think you guys all have this down. You answered that question on the quiz about sputum wonderfully, but just to review it again, it's making sure it's sputum and not spit from their mouth. So we want to see less epithelial cells, so less than 10, and lots of neutrophils to indicate a bacterial infection. Cerebral spinal fluid cultures, the main disease here was meningitis. There is an aseptic versus purulent. So aseptic was more of viral meningitis with increased lymphocytes. Purulent was your bacterial meningitis. So purulent is much more significant and severe to the patient than aseptic. I mean, meningitis is never a good thing, of course, but out of the two, bacteria is the much more scary one. There is, of course, fungal meningitis, parasitic meningitis, but these are the two that we talked mostly about. With the purulent being bacterial-based, so you would see increased neutrophils, increased protein, and a decreased glucose results in that cerebrospinal fluid sample. Most common cause in children under the age of 6 was Haemophilus influenzae type B. Again, they now have a vaccination for this, thankfully, so it pretty much has really lessened the amount that we have seen of Hib causing meningitis in children that young. Neisseria meningitidis, strep pneumonia are still big guys causing meningitis. There is also a strep pneumo vaccine, and that's typically given to older adults. Neisseria meningitidis is more with like the dorm rooms type deal. I believe we discussed that. And then strep A galactiae was the one that we screened pregnant women for to see if they're carrying it in their vaginal tract. And if they are, we try to give them antibiotics, clear it out of them before they give birth, because otherwise they can pass it to their newborns, and then it results in a neonatal meningitis. The other one that does this is Listeria Monocytogenes. Remember, that can also give neonatal meningitis, but you usually get that more from different foods that you eat, like deli meats, homemade cheeses, soft cheeses, things like that. So they just have to be careful of what they're eating while they're pregnant. So with the sample collection, it is a lumbar puncture, and we collect different tubes and we send them to the different areas of the lab. So tube 1 should always go to chemistry. Tube 2 then is going to microbiology, because hopefully if there was any skin contamination, it was seen in tube 1 and it wouldn't affect our microbiology sample. Tube 3 is hematology for cell counts, and tube 4 is our other, sometimes sent to cytology if we need it to be. Other things that we discussed briefly was a stain called India ink. This is specifically used to screen and detect Cryptococcus neoformans. That is a yeast that is seen a lot with AIDS patients or other immunosuppressed too. And so with the India ink stain, basically it stains everything in the sample except the Cryptococcus neoformans. Cryptococcus neoformans will be highlighted or kind of look like a halo because it has a capsule, and the India ink cannot get into the capsule. So everything else will be like a brown look except the Cryptococcus. Eye cultures, Blepharitis was just a brief mention in that, which is the inflammation of the eyelids, and that's mostly caused by Staph aureus. Otherwise, the biggest eye disease that we discussed was conjunctivitis, caused by lots of different organisms. Some of them are listed here. So you got your couple STDs, your chlamydia and your gonorrhea, and then Moraxella, Strep pyogenes, Haemophilus influenzae aegypticus. That last one is the one that causes pink eye a lot. So basically, you have your same type of organisms we see over and over in the upper respiratory area. Moraxella, we talk a lot about. Staph aureus, we discuss a lot. Streps. See, we kind of get used to seeing the same guys over and over here. Ear cultures, you have your external ear infection. Gosh, my brain just went dead. So we commonly got a swimmer's ear, especially caused by Pseudomonas aeruginosa. It loves warmer waters. We talked about hot tubs with that. You got your middle ear infections, otitis media. That's the more common one that we see. So again, Strep pyogenes, Moraxella, Staph aureus, Haemophilus influenzae. So the same little markers over and over again here. Your sinus culture, Strep pneumonia, Staph aureus, Moraxella. I mean, it's a big deal. And then GI. So with GI cultures, we discussed the 3 different categories of toxin. Please be sure to know these 3 categories, what they mean, and the example bacteria that go in each. So your enterotoxins is releasing all sorts of fluid and electrolytes out. It's a very watery consistency, so that would be cholera especially. Remember, cholera has those characteristic rice water stools. So that's a great example of how that enterotoxin works. We also have Enterotoxigenic E. coli. The beautiful thing there is it's named for the toxin, enterotoxin. So that's your traveler's diarrhea. And then you have Shigella dysenteriae and Salmonella. Cytotoxin is where it actually affects the cells, cyto for cell, so slots the cells off, leaving unprotected surface. Clostridium difficile, Shigella, enterohemorrhagic E. coli. I think we have hammered home the enterohemorrhagic E. coli 157 in the discussions a few times. I have no idea how many times that was asked, but you guys should be experts at that now. And then neurotoxin was more food poisoning, Staph aureus, Bacillus cereus, Clostridium botulinum, and perfringens. We discussed H. pylori, Helicobacter pylori. That is the main cause of your gastritis or your ulcers. The big test there is that it's hugely urease positive, so it will show up a really bright pink on that test showing a positivity. Other pathogens that we just always screen for in stool samples. So anytime we get a stool culture in with complaints of diarrhea, bacterial-wise, we're immediately looking for E. coli 0157, not just normal E. coli, just certain strains of E. coli, because otherwise E. coli is a normal flora of your intestinal tract, so you will see it, but we're looking for certain strains here. Salmonella, Shigella, of course, and Campylobacter jejuni is the other one. Don't forget about that. That is a bugger that we see quite frequently causing diarrhea. That one, remember, wants a little bit of a warmer temperature at 42 degrees Celsius. And then, of course, the other thing that causes lots of diarrhea is parasites. We also do screen for parasites too, but we're talking more bacteria here. And then your skin and wound cultures, first and foremost, again, normal flora of the skin. We already discussed this with the blood cultures, but this also affects if you don't properly collect the wound culture. If you're not doing a sterile collection, you can easily pick up these contaminants and they will grow it on the agar later. So again, Crinibacterium, Bacillus, Staph epi, Propionic bacterium, acnes, all normal flora, so all can be seen as contaminants on the wound culture. If they collect the wound culture properly and with a sterile technique, typically you'll only find a pure culture growing on a wound. You should only see one organism, unless, God forbid, they might have a double organism causing it, but really unlikely. It's usually just one. So you should see a pretty pure culture if they collected it properly. So common skin wound culture diseases, cellulitis, again, that's more the, not skin surface, but under the skin, like a subcutaneous maybe infection, a lot of times caused by Staph aureus. That's something that they take really seriously. We don't want that to get systemic. A lot of times they might be put on IV antibiotics, not just a pill, but an IV antibiotic to help get rid of it. Necrotizing fasciitis is, of course, always horrific and serious. Thankfully, it's not too common, but that's the eating through the layers of the muscle fascia. Strep pyogenes is a huge cause of that. Folliculitis is inflammation of the hair follicles, Staph aureus again. And impetigo, which is a lot of times seen with babies, is the classic sign of impetigo is like by the mouth. It's like a round circle right by the mouth. That's Strep pyogenes as well there. So it kind of goes every other one here. And then, of course, wounds. We didn't mention that specifically. You can have a wound anywhere on the body, depending on what happens to the person. And really, almost anything, again, can infect the wound if it got exposed to it. Again, there is one that we see commonly with it, but there's just so many different organisms that can cause wound infections that it's too huge to list out. All right, so that should be everything to help get you prepared for the exam. The exam itself, very short. I think if I remember correctly, I just looked at it this morning, it's only 33 points. It's kind of like a large quiz. And most of it's multiple choice. There is a matching question, and there's a short answer that you guys should easily be able to do the short answer. So yeah, really, I expect you guys to do awesome. So that's it. Have a fabulous week. And as always, if you need anything at all, please feel free to reach out. Thanks.